specific question before we start?
I have one question.
Yeah, go ahead, please.
So in the PowerPoint, you talked about the virus being a parasite, but
Yeah.
can't the term parasite only be used with organisms?
For living organisms, absolutely. You are 100% right, 200% right. And this is one of the problem that even the textbook writers, they do actually say that we should not, and there is a slide that you will see that we try to avoid these terms which are, we call anthropomorphic, which is related to any kind of living organism related to, and we just can't avoid that. I mean, like whenever we use the term replicate, reproduce, propagate, all these terms are related to living things. So you understand what I'm trying to say?
Right, I understand. Yeah. It's just, it was like in one part, there was like, oh, they're not living, and then
we are trying to say it a parasite. Yeah, exactly. That's what it is, and that is the best definition that we can come up with if you just look around. But really, the argument in correct word, you can absolutely argue that we really cannot call it parasite in the sense because it's not living. But unfortunately, what happened that when it goes into the cell, it becomes it goes into an active form, controlling the cell. At that time, its only purpose is to propagate itself, so it's get benefited, whereas the cell actually get harmed. And that relationship, if it was a living organism, is absolutely parasitic. But unfortunately, we just cannot. We just do not have a term that can replace it, or we have to say the whole sentence that the viruses will go into the cell, and it will each time we have to tell the full sentence that it actually benefits itself and harm the host. So that's the reason that probably we just replace that just for our convenience. But in real matter, when we talk about that, in reality, it should not be used on a term when it's there. Yeah, so there could be absolutely, you are correct. Yeah, we can argue with that.
All right. Yeah. One thing I did see before for like to not
anthropomorphic. Yeah.
Yes. Was it competes with the host mechanism.
Yeah, that's a good question. You can just absolutely, you can do that, but the thing is that what happens that you can just use that's a good question, competes with the host mechanism, but what happens that it just tells you that what its purpose is, but each time it actually wins completely. So because each time it just outcompete the host pretty much all the time. And so yeah, that's a good term. Yeah, absolutely, you can use that as well. Yep. A good point. Yeah, these are very good suggestions as well. Yeah, absolutely. Anything else that you guys have? Good. All right. So let's start. What we will have, you will see that I work on these PowerPoint slides until the last moment. And some of these, you will see that notes have been updated, and those. So just what you do, that's just rewrite your PowerPoint slide that you have downloaded earlier, and you will have your notes. It's the exact same file name. Most of the PowerPoint slides has not been changed other than just a few figures or something like that. But other than that, they're all good. So let's start. So first we are going to see that what is a virus, and it's a pretty easy chapter. We are not going to see too many supplementary material that is not needed for this chapter. That's why I just did not give you any additional notes. The notes below the PowerPoint slides are sufficient for this chapter. And what you're going to see that after we finish the class, there will be a quiz, which will be due Sunday at 11:59 p.m. on this chapter. That will be up. And again, it is unlimited times, and you have 3 times you can attempt it. So we will start from there. So we have already talked about the primary textbook sources used in this course. Principle of Virology, this is the book which we will mainly follow. And again, I said that one of the book author is Vincent Racaniello. He is a Higgins Professor of Microbiology and Immunology at the Columbia University School of Medicine in New York City. And this course is designed pretty much following his course. And he gave me a lot of inputs about this course as well. So very, very similar. Another book that I use is the Fields of Virology. We will use that one, and the Prescott's Microbiology. This is a very, their virus chapter is also, I will take some of the pictures in there. We'll see that. All right, so the first thing that what we're going to talk about, that we live and prosper in a cloud of viruses. And viruses infect all living things and everything. I mean, like each one of us is actually infected with the viruses. We regularly eat, breathe billions of virus particles. And the interesting part is that at this point of time, it's kind of funny, all of us are infected. And in fact, probably this infection of these by the viruses happened in utero when we are in the fetal form in there, and just remains as a part. And this is just like the microbiome that we have. We know that our gut is actually colonized when in utero, the same way we do have a virome, and we are just starting to learn about that virome. You always hear about the microbiome all the time now. Very well studied, a lot of people, and we are just started learning, and there is a whole new field out there about the virome, but we regularly, we are all infected with some of the viruses, and it's kind of like, in a present day, it's kind of really alarming. Is like, what do you mean by we are all infected? Yeah, we are all infected. There are some kind of virus in our body, and most of them actually are not doing anything. They are just passive agents. And another very interesting thing is that we carry the viral genome as a part of our genetic material. And the mind-boggling part is that out of our whole amount of DNA that we have in our, like this is for the humans, that about 1.5% or 2% sequence of our total DNA that we have actually produce functional proteins or functional products. And there are many other different things, but there are about 40% or more what we call as a retro elements. And these are the remnants of different viruses, mainly the retroviruses, which infected human beings over millions of years, and it became a part of it. It actually does not produce any viruses, but it does control some of our functional genes, actually. So that's the interesting part. And the viral genome integrated in our genome is a kind of a molecular clock. So if you just actually get a fossil from somewhere and you just get some DNA and sequence it, you can actually tell by these signature viral sequences in a genome of, say, dinosaurs or something like that, what is the approximate age of that nucleic acid. So very interesting. And they are mainly passive agents, and many of them actually don't do anything. So you can just easily look at that. And one of the things that I actually, in the notes area, I just for your interest, I just put a link in there that there is actually something which is called the ERVs. And these ERVs or ERVs, these are called endogenous retroviruses. And this is what I'm talking about. These endogenous retrovirus sequences are the integral part of our genome. And it has been clearly shown that there are some of the gene functions, and particularly their expression, some of the corticotropic hormone expression actually is controlled by these retrovirus elements or these endogenous retrovirus sequences. And you can just look at that just for your interest, not needed for your exam or quiz, that how this controls. So just being said that virus and all other organisms evolved through the evolution at the same time, hand in hand. So that's about it. So the number of viruses on the earth is actually kind of mind-boggling. And you can just see that there are more than about 10 to the power 30 it's very difficult to wrap your head around that number, even bacteriophage particle in the world's waters. Now, we are not talking about the space or the atmosphere and the land, just only the waters. And just think about this. This is interesting that a bacteriophage, which actually infect the bacteria itself, and there are many different types, 10 to the power negative 15 grams, so about a femtogram. And if you look at that number, 10 to the power 30 multiplied by 10 to the power negative 15, the biomass on the planet of the bacterial viruses alone exceeds the biomass of elephants by more than 1,000-fold. I'm like, that is like really amazing that what you can think about, that even with that femtogram number, just the sheer number is mind-boggling. And this is something that you can just actually look at that link in there, that if you just take these 10 to the power 30 bacteriophage and put them like tail to tail, you just put another one on top of this one, and then put another on top of this one, head to tail, then the length will be 100 million light years. So I mean, like, it is really, these are some of the amazing facts that you have. And none of these facts are, you just do not have to memorize these numbers for that, but just for an introductory idea that these are some of the amazing facts of about the viruses. So here also, another very important things, that although we are always thinking that at this point particularly that the viruses are always bad, but they are always not just purveyors of bad news. So you can actually think that how, and we're going to argue that why not, that if you just look at the biomass overall, you can see that the prokaryotes right here is the majority. These are the protozoan or the protist, and these are the viruses. But if you look at the abundance, this is 94% of the biomass is viruses, but the mass is, or the total number is, the abundance is viruses, but their biomass is much less because they are much lighter. So interestingly, more viruses in a liter of coastal seawater than people on Earth. So it's kind of like when you just swim and just take a mouthful of seawater in your mouth or something, like spit out to your friends or something like that, you are spraying like millions of viruses around. And we are perfectly fine with that. These viruses do not do anything, because many of these viruses, like the reason that why these viruses don't cause anything, because these viruses probably infect something else. They are not our viruses. They are host specific. Some of the bacteriophage viruses, bacteriophage, they do not infect us. So that's the reason that we just are absolutely fine and they don't do anything. And the most important part is that these viruses actually do kill a large number of prokaryotes. And while killing these prokaryotes, that means they just lyse them. And these prokaryotes actually sink at the bottom of the ocean, and the biogeochemical cycle drives the ecology or keeps the ecology in balance. So they are also a part, like the viruses are also the part of the biogeochemical cycle to keep our planet in balance. So very important compared to the other ones as well. So this is another interesting thing, that the virome, and you can see that if you just look at that your skin, hair, nails, these actually mainly contain the DNA viruses. But if you just look at that, and we will know that the viruses can have a DNA genome as well as an RNA genome, and digestive tract, blood, urinogenital tract, we have not isolated any RNA viruses from there yet, and also from the skin, but all of the others also do have both type of viruses. And we are just learning these about this. And the way we are learning is now you know that you can just take a sample, and this is what we call as a next generation sequencing by pyrosequencing. There are many different techniques that you can just Google that the word, that next generation sequencing and how they do it. These are high throughput sequencing. You can just sequence from a very little amount of DNA or RNA material or you can do the RNA where you can sequence the RNA as well from a very little amount of starting material. And we call this as a deep sequencing, where you have very less number of copies of the genomes. And we just isolate these samples from these different tissues and just randomly do these sequencing, and we are discovering viruses this way. And we call this one as the metagenomic analysis. And how people do this is a very interesting way to do that. That this is actually in the off the coast of Australia, and these are some of the humpback whales. And what they are doing, these scientists, they do have a vessel or a ship, something like that, and they are actually sending out a drone which they can control from the vessel or the ship. So what they're trying to do, that they are looking at the viruses, what the viruses present in the whale breath. So what they do, that they send this drone out, and you can see that these whales are actually expelling their breath, and you will be able to see that how they trap this vapor inside this container and bring the vessel back. So let's take a look at this. Okay, all right, now take a look at it. So you can see that the drone is opening up that right here. So just to have an idea, that's just the way that the scientists actually go and collect these things. So this is basically the whale breath virome that they're studying. So a very interesting way that this is a whole new area of the virome research that is opening up. We already know that the microbiome, but we had just started learning about that. So there is a lot to learn about this as well. So amazingly, the vast majority of the viruses that infect us have little or no impact on our health or well-being. And we are going to see that why it is so, that why not. The reason, one of the most important reason for that is many of these viruses that are inside our body, they are passive agent in the sense that they are specific. And what we mean by the specific, that they are specific for a different host. So many of them are actually agents, infectious agent for something else, and they're just passing through it. They do not have the capability or the ability to infect us. And that's the reason that majority of these viruses actually do not infect us. And most of the viruses that actually pass through us is, interestingly, are the plant viruses. So we just consume them along with our salad, in our spices and everything, fruit, vegetables, all the time, and they just pass through us without affecting anything. So that's the reason that majority of them have little effect. And here, this is actually an array of cartoons from the Principles of Virology, that textbook. And you will see that you will get familiar with this cartoon, and you will understand that what are these different viruses. You will be able to tell them just by looking at these viruses, that what are they gradually at this point. So can anyone here point out or just tell me that can you identify any of the viruses that you know of in this picture just looking at them? Can anybody tell? All right, which one is the coronavirus? That's an interesting question. Can you tell? Like right on the screen, top of the screen, right, left? No question.
Is it the bottom?
Yes, this is right here. Yeah, good. Yeah, right here. This is the coronavirus, that what you are seeing. This is the cartoon of the coronavirus. And you can see that right here is the protein, which is a spike protein. And if you can see that there are 3 lobes of them because it's a trimer. Along with that, there are some other proteins like an M, or the membrane protein, and an envelope protein. There are 2 other proteins on the envelope. You have an RNA genome in there, and that's about it. So you have an adenovirus right here. You have some poliovirus right here. And you have some measles virus. This is the variola, which is the causing of the smallpox. Right here, you have the influenza. You have all these different viruses, HIV right here. This is right here. This virus is a baculovirus. It's an insect virus. And gradually, you do not have to remember any of these, but all of these cartoons are actually drawn from the real structures that you have, and you will be able to see some of those. Say, for example, this is a bullet-shaped virus. This is a rabies virus actually look like that. And you're going to see, particularly, I just want you to pay attention to this. We will see some electron micrograph of this particular virus right here, the rabies type virus. There are many other viruses which looks like this as well, but you will see that it's remarkably similar to the real virus or the electron micrograph. So here, the reason that why most of the viruses actually don't do anything to us, because most viruses just pass through us. We ingest many of these non-animal viruses regularly with fruit or vegetables, and as I said, and here is actually a study. So you can see that right here, this is called a cabbage looper. And these cabbage loopers are infected with a virus almost always, which is an insect virus. It's a baculovirus. And if you have studied some of the papers or something like that, right now, this baculovirus is actually a molecular biology tool for expression of genes or heterologous proteins in insect cells. We actually use genetically engineered baculoviruses to produce some of the protein in insect cell culture, highly used in biotechnology and pharmaceutical companies to produce even some of the products of need. So basically, almost all cabbage that we get will have some of these baculoviruses. So you can see that such an extent that about 100 square centimeter of leaf material would contain about 10 to the power 8 particles of the virus, which is pathogenic to the cabbage looper. And we are just constantly, say you are eating coleslaw, you're just eating viruses all the time. And that is perfectly fine. It doesn't do anything. On the other hand, you can see right here the metagenomic analysis that what we were talking about. The RNA viruses in human feces reveal most of the viral sequences are similar to plant viruses. And that's the reason that they just pass through us. They don't do anything. And you can see that many of these metagenomic analysis of the shows that 91% of the sequences that we actually get actually resemble plant viruses. And one of the interesting part is that most abundant human fecal virus, and if you just take a stool sample and send it to a sequencing place and they sequence most of these deep sequences, metagenomic analysis, if they do it, and if you just look at the abundance of the different virus, this is the most common one in human fecal matter, which is called a pepper mild mottle virus. So you just, this is like in the black pepper seed, basically, that you grind up and add as spice to all for your food, and you just ingest this pepper mild mottle virus all the time. And this is kind of funny that what I thought, that most of them are passive agents. All right, so does virus good something to someone? And at that point, what we have that we may have to, because initially someone told me that we use this anthropomorphic term, which is parasite, which is not correct. On the other hand, they compete. Another term that you can use is they compete for the mechanism for survival with the host. That is absolutely correct. But we may even have to rewrite that statement that was true for years, because we are just started to find some of the viruses which may be good. And they then are we going to tell that viruses may have all kind of relationship, like they help each other, they are symbiotic? Maybe at one point of time. Not at this point, because we still think these as exceptions. And at one point of time, we may have to rewrite the definition of viruses. So let's see that what this does. I actually made a correction in the notes of this. So this is actually a grass. These are really, really tall flowering grass. They can just grow 1-3 meters. And this is called the Dicanthelium lanuginosum. And some of the families of these grasses are found in the tropical area, which I wrote in that's a tropical grass, but this one is not a tropical grass. This is a native grass of North America, and this grows in the geothermal areas of the Yellowstone National Park. Very, very large. It's like if it grows fully, it's taller than a human being, 1 to 3 meters, as I said, 6, 7, 8 feet tall. So what happens that they grow in an unusually high soil temperature. You can see that about 55 degrees Celsius, because they are close to many of these hot springs and geothermal areas in the Yellowstone. Now, these grasses are colonized by a fungus which is known as the Carvularia protuberta. This is a fungus that lives in association or symbiotic relationship with this grass. Now, this particular fungus is actually infected by a virus. So you can see that this is called a Carvularia thermal tolerance virus, CTHTV, the full name, Carvularia, the name of the fungus, thermal tolerance virus. So this virus infects these fungus. This fungus lives in association with this Dicanthelium grass, and it allows it to grow or allows its thermal tolerance. So what happens that if the fungus does not colonize into this grass, they don't grow. So this is a three-way symbiotic relationship where this virus is actually giving the thermal tolerance to this fungus. If the virus does not give if the Carvularia is not infected with the virus, it loses its thermal tolerance. Only if the virus infects the Carvularia, it can withstand that temperature. At that temperature, it can colonize into this Dicanthelium and allows it to grow. So you can directly look at this paper in the Science magazine. And this is probably a three-way relationship in plant, fungi, and viruses, and one of the good viruses that we can think about. Another example of this is that we have just started to learning about this, and this is also a Nature article that you can look at, that there is a norovirus, and this is the murine norovirus. So this was basically studied in a mouse, murine, and this norovirus actually caused gastrointestinal disorder in human beings. That's a different strain. This is a different virus, but in the same family, the norovirus, and this is what we call as a cruise ship virus. Sometimes gastrointestinal disorders or diarrhea actually breaks out in cruise ships, and those are caused by the norovirus mainly. But that's something different. So what we are looking at here, that this enteric virus can actually replace the beneficial function of the commensal bacteria. So what we are looking at in this diagram. So first of all, what we are looking at right here on this side, we have some healthy mouse, and they are stained by hematoxylin and eosin. This is a typical dye that we use for histology. And you can see that the cytoplasm is stained as a pink and the nucleus is blue, you can see. And these are the villi which are lined by these epithelial cells. So each one of these finger-like villi. So this is the finger-like projections which is projected towards the lumen or the hollow of your intestine. And this is just a close focus of one of these, and you can see these individual cells. And you can see that they are very healthy, fresh looking, and they are lined by what is called as the brush border. All of these epithelial cells do have a brush border, which is made up of microvilli. So remember that there is a difference with that. This villi inside the intestine is made up of thousands of individual cells, which are each one of them, thousands of these individual cells make up this villi. This increases the surface area of the intestine. And each of these cells actually do have these microvilli projecting out of them, and they help in the absorption of the food material. And this is very healthy. And this is, on the other hand, is another stain. And this is some of the cells are being stained by an anti-CD3 antibody. Now, CD3 is a co-receptor in the T cells, And so what you are seeing, that these brown dots that you are seeing, these are the location of the T cells. And why you have the T cells in here, these are the lymphocytes. They are present in the villi, so that if any infection actually happen, any bad bacteria or anything or viruses actually enter into the intestine or into tries to come inside the body, these T cells will actually attack them. That's why you have a healthy population of the T cells. Now, what you are looking at here, the GF means this is the germ-free mice. So the germ-free mice are actually immunocompromised, and at that time, the germ-free mice don't have any kind of bacteria in their gut. And you can see that as you do not have any commensal bacteria, the structure of their villi is not healthy. You can see that if you just compare to that, they are broken up. And you can see that how the number of cells and the way it looks very sickly right here compared to the right convention or the conventional mouse, which is the healthy mouse. And if you look at the stained CD3, you almost don't have any T cells in there. So just maybe a little few right here. So this is definitely a condition where these germ-free mice can sustain very severe type of infection if exposed to the normal environment. Now, when these germ-free mice were added or fed with this murine norovirus, you can see that it can actually restore the condition. So you can see that now it is become much more healthy. They are much more fatter right here. These villi, the villi right here, and you can see their structure, and you can see that now you have much more number of T cells that came into this villi, which will protect these intestinal things. So basically, what we are trying to say here, that these murine norovirus is actually helping the commensal bacteria and the gut morphology to go back to the normal thing. So this is one of the first observation that maybe this is the virome of good viruses can help us, and we are still learning about this. All right, any questions so far? Good. Okay. All right. So next, what we are trying to do, that the viruses are actually amazing molecular nanomachines, and the reason that we call this as a nanomachine, because they are not the living things. We are going to come to the next part explaining about that thing. And what we are looking at, that these are some of the micrographs from cryo-electron micrographs of some of the viruses. And basically, they are just made up of some of the macromolecules, and these macromolecules actually do some chemical reactions to do all these amazing things that they do. The infection process and everything is just a chemical reaction. They are just do not have any brains or anything like that to strategize anything. It's just the evolution of some of the chemical processes that actually does all of these just the way it is. And the virology is an integrative science. And why this is so, that this is the virology is just not within the biological sciences or microbiology or molecular biology itself. To study the viruses, you need to, many of the physicists actually are study the virology. The reason, the whole of the imaging process, that electron microscopy or cryo-electron microscopy, or when you are looking at the molecular structure of the viruses, that needs your knowledge of complete physics and chemistry, and particularly that area of the viral structural biology is mainly the scientists are from a physical science background. They are coming into studying the viruses rather than biological sciences. And then you have, as you can think about that in the context of this pandemic, how many different disciplines are involved in that, from epidemiologists to social scientists to the doctors and everything, just due to this, just not only for studying, but when you have a viral pandemic or any kind of disease, there are so many different disciplines actually get involved in studying this. This is true for many other things as well, but that's the reason that we call this the This is a very integrative science. You will see all types of people from different disciplines. They are involved into the virological studies in many different ways. Environmental studies, say for an example, we are constantly measuring the amount of the viral RNA in the wastewater at Clemson. And these are done by some of the scientists who are not in the biological sciences department, but this professor is from the Department of Environmental Engineering. So, and civil engineering, they are actually trying to do these. So very integrative science, you can think about that way. All right, so the course goal is designed to help you see the big picture of virology. So the other way, the virology, or classically a lot of people used to teach the virology, is we start with a classification of the viruses, and we call this is like, okay, all right, these are the animal viruses, these are the plant viruses, these are the bacterial viruses, and each day we just come up with that, okay, all right, you know about poliovirus, herpes virus, this virus, this virus, this virus. We just describe the virus and we go home. This is not needed in 2020 because all this information is at your fingertips. This course goal is to give you the big picture of the of virology, in the sense that we are going to tell you that how the principles of the virology, that how these things actually do work. Most of them do have a very common way to work, and that's what you are going to learn. Once you learn that, you can actually understand each of these viruses, and you can just read it from a literature and understand how they works. So that's the reason that we are teaching this as a principles of virology, rather than just information for an memorization of 100 different viruses. So that's the difference, that how the virology used to be taught earlier compared to now. So again, remember that. You will also understand that how to think about virology as an integrative discipline and not an isolated collection of viruses, diseases, or genes, that is, as I said, and learning the fundamental about these molecular wizards that will amaze the informed and frighten the uninformed. And you will be informed completely after you have this this course, and you will enjoy that as well. So you can easily see that this is what we call as a BSL-3 laboratory, where you have this spacesuit like your personal protective equipment, PPE, and you have air coming into your. You are just completely sealed in there, and some of the this is what is called a BSL-4 or Biosafety Level 4 facility, where you have your own air supply. And for some of the agents, you need this kind of facility. There are several. We are going to talk about these. So that's the overview of that. So this is also, I actually directly took this slide from Vince Ricaniello's slides. And this is probably 2009 when this H1N1 actually broke out. And this H1N1 influenza was the same strain as the 1918 flu, but it did not cause a pandemic in 2009 because we were much more informed, and we had influenza vaccines, some of the drugs as well. We know that how to isolate people. So not much happened, but still it was at the level of a pandemic to some extent. But see that when the media actually do report, they actually definitely read a scientific publication or something like that. But how they portray it, just see that. Ravage the lungs, okay, all right. Spreads through respiratory system, cause lesions, that's fine. Doesn't stay in the head like seasonal flu. I mean, like, so this is kind of mind-boggling. Where in the world influenza goes into your head? I mean, like, influenza does not go into your brain or the head. It goes into your upper respiratory tract. It infects the lungs. So these are some of the things that does not stay in the head. We call this as a head cold. And from that, you can see that this is the way that the media portray. And you will see that there are many of these type of things that actually comes in there, and you are going to debunk all these and correct these things as well. And this is the sad part that I just had to write this, that the sad and the scary part is that even the doctors and scientists have misconceptions who are not specifically virologists sometimes. And we are going to have almost a fight. We are going to just have a huge discussion about this even later in the course, but we will start that discussion to some extent today as well, and we'll see that why we say so. We'll come to that slide. So first, let's start with some of the definitions of the viruses. It's an infectious, obligate, intracellular parasite, and again, as somebody said, that it is also a controversial term, comprising of a genetic material, DNA or RNA, surrounded by a protein coat, sometimes a membrane. And I have given a considerable amount of notes on this because you want to dissect that. So the first is the genetic material for all living organisms is only DNA that we know of. But this is the first time that you are looking at something, an agent or an entity, where RNA is the genetic material as well. And I explained that why RNA is not a genetic material for other organisms, because there is a limitation that how big an RNA genome could be. Approximately, it's about 30 to 35 kilobase. If a genome is more than 35 kilobase, it is very difficult to keep the RNA genome stable. And what we mean by the stable, that in the environment, there is an enzyme, which is very simple, that digests the RNA, which is called an RNase. And this RNase is everywhere. And you know that you on your palm, on your hand, you are producing RNase all the time. So if you work in a laboratory which actually deals with the RNA, you will actually understand that how crucial it is to handle your RNA to keep it out of these RNAs, because it's going to digest and all the time. So that's one of the reasons that in the environment, RNAs are always susceptible. And the viruses do have a way to protect them because they actually cover the RNA with some proteins, even inside the viruses as well. But still, it is much more susceptible to the RNAs as such when they are being propagated. And that's the reason that sometimes we think that they have converted into the DNA to make much more stable viruses. So think about that. All of these viruses do have a DNA or RNA in the middle. And that DNA or RNA is covered by a protein coat. And we are going to talk about all these terms and names as such. And they are covered by a protein coat. And then outside the protein coat, you may have a membrane as well. Not always. And I just compared that think about a present inside a cardboard box. So the present inside the box is the genetic material. Your cardboard box is surrounding it. And sometimes you may have a wrapping paper on the cardboard box that will be your envelope, which is a phospholipid membrane. And this membrane is almost always derived from the host cell as the virus is coming out. So let's see right here, you have then HIV, which does have a membrane outside it. As the HIV buds out of the cell, it steals this membrane from the cell that it is budding out. Now, think about this way again, that I also wrote in the notes, that sometimes you have the wrapper around the box of the present, and you just attach bows or stickers and different things on top of that to decorate it. So what happens, the virus actually decorates, most of the viruses, this membrane with some of the viral protein. And right here, you are seeing that this is a protein that's being decorated by the virus. Let's take a coronavirus for an example. So right here, what you are seeing in the middle of the coronavirus, it has an RNA genome, and we will know that it's plus, minus, and all those things. And outside this, you have a protein coat along with that. And then you have a membrane, that light brown right here in the membrane. And that membrane is decorated by all these different proteins, including spike protein, and there are two other, which is called the envelope protein and the membrane protein, E and the M proteins. So you can see that. So compared to these different types, so you can see that most of these are envelope viruses, but right here, this one, right here, this one, this is a picornavirus, like a poliovirus. This is an adenovirus. These are naked viruses. This is also another one, which is the reovirus right here. These are not enveloped. These are the naked viruses. So the naked viruses is that inside this, you have the present, and you just have the cardboard box that you received from Amazon. Just looks cardboard and written as Amazon Prime. That's it. So those are your naked viruses. If you actually tell Amazon to gift wrap it, they will wrap it, and that will become your enveloped viruses, which are mostly like those. So what you are seeing, that most of the viruses here that is being shown are enveloped viruses, but again, there are lots of naked viruses as well. And again, to dissect the word that obligate intracellular parasite, that they have to be inside a cell to replicate or propagate. And again, replicate or propagate is a term that we will use for an organism, but again, we are just sometimes have to use some of these terms. So without a living cell, there is no entity of any virus whatsoever. So that's the reason that we have to have a cell to replicate. Say, for an example, if you just draw some blood, you may find an infected person do have viruses in the blood, but in the blood, that virus cannot replicate. It does have to go into a specific tissue or maybe even a blood cells, like say, as I said, HIV actually infect your T cells, CD4 or the helper T cells, but you need a cells, even if it is a blood cell, the virus needs to go inside a cell to replicate itself because it's dependent upon the cellular machinery. It will not replicate in a broth, tissue fluid, or just plain blood. So that's the reason they're obligatory, absolutely needed intracellular parasite. All right. So next, what we are looking at, that these viruses are obligate parasite, and every solution must reveal something about the host cell as well as the virus. So this is just an overview of that what we are going to talk about. Many of the cellular processes that you study in your cell biology or microbiology somewhere else, all were revealed by the viruses. And the reason that is amazing, because these machines can be traced, so you can infect the cells and look at that, what are the things happening, and you can actually understand that phenomenon. And that's one of the reasons that we understand these cellular processes is due to the viruses. So here what you are looking at, that some of these viruses, these are actin filaments. And if you have taken cell biology or will be taking cell biology, you will understand that how these actins are remodeled by different processes, how the extracellular matrix actually attach the cells to other. And all these functions were initially actually studied through the viruses, which actually disrupt these processes or enhance these processes. So that's the reason that we call that every solution must reveal something about the host as well as the viruses. All right. So now some of the definition that the virus and the virion. So the virus is an entity made up of many macromolecules. And what we mean by a virion is an infectious virus particle. So here you are looking at a molecular model of a virion. And this is not a cartoon. This on the right-hand side is these are cartoons, but this is actually a three-dimensional model, which is modeled after a cryo-electron microscopy. So these are the virion, infectious virus particle. And again, you can always argue that we are constantly doing this, that why virus is not an organism, that we can argue all throughout this course as well. So here, what we see, a typical viral life cycle of an infected cell, and we will talk about each one of this in subsequent chapters. But what we are seeing, that what is happening right here, that the virus, the first part is entering the cell. But again, you do not have to know each step right now, but it has to attach it with a receptor. Sometimes it needs a receptor. Sometimes a virus can fuse with the plasma membrane directly, and then it can just travel through the microtubules or others into the cell. It can just go into the ER or some endosomal structure, and these details are not needed. And then finally, it uncoats and deliver its nucleic acid inside the nucleus most of the time. And once it delivers the nucleic acid in the nucleus, it will make the mRNAs. These mRNAs will come into the cytoplasm and make viral proteins. And again, it will make more mRNAs, some early, some late. And along with these mRNAs, you can see that all these virus proteins are being made. These actually make up the box of the virus. But you need the present, which is inside, which is the nucleic acid. So this is the original genome of the virus. It needs to replicate its genome. So one genome goes in. If you want 10,000 virus coming out of the cell, you need 10,000 copies of these genomes as well. So you will copy the gene, and then you need the box material to cover these genomes. So you made these proteins. So these proteins altogether will cover these to make the whole present, and these viruses will exit. This process is called the assembly. And after assembly, the virus will come out of the cell, which we call as an exit. And we will talk about detail about these. So this is, in this process, the virus is replicating inside the nucleus. Remember that some of the virus nucleic acid don't, particularly for the RNA viruses, they don't have to even go into the nucleus because they carry their own enzyme to replicate in the cytoplasm itself. So in this case, it's just being shown in the nucleus, but it's not all of the case. So that's your typical viral life cycle that we are going to talk about. And here, this is, be careful to avoid anthropomorphic analysis or expression, but it is almost impossible to do that. Viruses do not think, employ, ensure, exhibit, display, etc. But we will try not to use, but we sometimes just can't. They do not achieve their goals in a human-centered manner. They are passive agents. They are not intelligent entity and trick us. So think about that. A viral infection is a series of chemical reactions that randomly took place to make it happen completely by chance and selected and stayed over the years by viral evolution. So basically, just think about that coronavirus that we're talking about constantly, that they will just fall on some mucous membrane somehow in your nose or mouth. And it is just completely by chance that it will bump onto a specific receptor, which is an ACE2 or angiotensin convertase 2 receptor. And once it touches the receptor, the cell will take the virus inside it. And that's how it all starts. So it is completely by chance process. So they are incredibly small. You know this very, very well. So here you can look at that. This is right here is a bacteria. This is a red blood cells compared to an E. coli. And these are the bacterial ribosomes. And this is one of the largest virus, smallpox virus, which is the variola. Poliovirus, about 30 nanometer. Bacteriophage, 24 nanometer. This is your tobacco mosaic virus, about 300 nanometer in length. This is a T4 bacteriophage, which is a large bacteriophage. So you can easily see they are very, very small, even compared to the bacteria. And this idea is now we are rethinking this because we have discovered something else. So we will come to that. So how many viruses can fit on a pinhead? So this is just some idea that what you are seeing, that this is just a micrograph of a pinhead. Right here, we are looking at this area. And just to compare that, this is just a dust mite that you have. This is dust mite, and this is a hair. And if you just take this area and enlarge it, and this is just, again, a cartoon, you have these red blood cells, some pollen, some of the yeast cells. And Right here, what we are looking at, these are some of the E. coli's. And right here, these are some of the viruses right here. Maybe some of the, as per the picture, it was written as that these are some of the Ebola viruses. These are some of the polio viruses. So you can easily understand that how small these viruses could be. So again, just an overview of the idea that a pinhead right here, this whole area, can contain 500 million rhinoviruses. So these 500 million rhinoviruses, these are actually the common cold virus. And when you sneeze, you fire an aerosol that contains viruses to infect thousands. So here we actually were talking about that we can just make a discussion in there that that's the reason that, but why we do not infect many of these people, what we think that we could technically infect. And that's the reason that we just argued all the time, because these are all chance factors. If they fall onto the plasma, on a mucous membrane, then they have to travel to the cell surface where they find the proper receptor, and then only they can get in. So you have to give a person a fair amount of number of viruses before they can get infected. So it is still a chance factor. And this is a strategy by the viruses by making so many of them so that that chance become more. So that's the basic strategy of viruses, why they make so many copies of them, unlike bacteria or fungus infecting other organisms. So we thought, and this is also another, that just in 2008, the first giant virus was actually discovered, and this was known as a Mimi virus. And there are many other names, and these are all basically discovered by many of the groups in France, and they named this Mimi virus, Mama virus. And you can think that this is a rhinovirus compared to a rhinovirus, or this is an HIV compared to that, how big they are. And you can easily see right here, this is actually a paramecium, and here you have a chlorella virus. This is a virus right here. You can see, compared to that, see that this hairy looking virus, and this is a Mimi virus. And these are the giant viruses, and we had to, again, at one point in time, we were talking about that you cannot see a virus without an electron microscope. And now we had to retract that statement, and we can see a virus with a light microscope. And this is amazing that right here, another giant virus, which is the Pandora virus, and you can see that this is a light microscope. They are about 1.5 micron in size. And you will think that this is probably a bacteria. So you can see the genetic material, and this is the protein shell around it. And these are the giant viruses. And there are many giant viruses that's being discovered, and many of them have been discovered in last few years. All right, so these are just some examples of the giant viruses. So just know the name of those, that's about it. So the next part is that the viruses hijack the cellular machinery to replicate or reproduce, and why they are different in terms of their replication or reproduction. Again, I'm using the anthropomorphic term, remember that. So if we look at the bacteria, so at 0 time, here you have a time scale in minutes and the cell number. So at 0 time, you are inoculating a sample with your bacteria. And you will have a small lag phase initially. At this lag phase, the bacteria is getting adjusted to the environment. And after that, it will divide by the binary fission. 1 will become 2, 2 to 4, 4 to 8, and you will have an exponential growth until a certain time where you will see that there is a depletion of media and production of toxic metabolites, where you will have a stationary phase, and again, you will go into a decline or a death phase, which is not shown. But basically, you will have a gradual logarithmic exponential growth curve. In the virus, you do not see that. So what happens that you inoculate the virus to the cells, and you will have a long lag phase, which we call, particularly for the viruses, as eclipse period. So at the end of the eclipse period, you will see that there is a burst of viruses, and lots of these viruses actually come out immediately by lysing the cell. Most of the time, there are some other where the viruses can bud out of the cell, but that's the process, because the reason for that is that the virus replicate by assembly of preformed components, which are their genome and the proteins into many particles. Make the parts first, assemble the final product, and then exit. And that's the reason that we see a sharp burst compared to the gradual increase in the number of organisms that are coming out. So these are the two main differences between bacterial growth curve and the viral replication or reproduction. So how old are viruses? And we will not spend much time. These are very self-explanatory. Say that this is a nautiloid cephalopod 488 million years ago. And we think that around this Ordovician period, which is greater than 450 million years ago, viruses probably evolved and likely originated before the cells. Now, this is also, again, controversial that, as we said, that they are obligate intracellular parasite. So how they can form before the cells? Now, think about that. The virus is a molecular machine in the sense that they are just an assembly or collection of macromolecules. And these macromolecules probably come together just by chemical means and combine into a virus. And these viruses just stayed there, couldn't do anything. And when the cells came in, they were able, or just happened to be, just by chance, could get into the cell and replicate, take over, compete, whatever you can think. But again, exact what happened, whether they were before the cells or not, we can speculate, but we really do not know. All right, so ancient references. Here you can see 700 BC, where rabid Hector. So this is a reference for the rabies. On the other hand, this is also, we are not sure, but this is from Egypt. This is 1500 BC. If you look at this priest right here, if you look at this leg, and it looks like that this leg actually suffered from a typical polio that we see. I mean, like, we do not know, but we think that this might be one of the case. This leg is normal, which is not. So maybe these are some of the early references, ancient references of the viral disease that we can see. Then immunization is an interesting thing, that even in the 11th century, the variolation due to the smallpox actually started in China. So what they used to do in China at that time, they actually used to take these smallpox pustules and directly inject that liquid into a person. And many of these people actually got smallpox and died, unfortunately. So the only reason that they used to do it, because they have seen that if a person actually gets smallpox, they don't get it again. And this started the variolation. This is not a vaccine. This was extremely dangerous and probably caused deaths of hundreds and thousands of people as well. Then in the 1700s, Lady Montagu, and she was the wife of an ambassador to Turkey, and that was the Ottoman Empire, and they actually started doing this vaccination type of things, taking these pustules and doing that. And she started it back in England, coming back. But at that time, there was no knowledge of this agent. There was no knowledge that it's virus. And then finally, 1790s, Edward Jenner actually did this experimentally, even without knowing that he's actually taking this cowpox virus from some of these maids. So remember that he did not inject the first person whose name is Peter with the smallpox virus. What he used to see, that some of these maids who actually work with these cows, they get a cowpox virus which cause some kind of blisters in their hand, but they were immune to smallpox. So he actually took this cowpox, one of these cowpox pustules, and put that into Peter and challenged him with the smallpox, which he didn't get. Like, you cannot do this this time. You will probably go to jail for the ethics. But at that time, that was the birth of the vaccination. You all know these stories. So the first virus discovered was the tobacco mosaic virus, and people did not like these blotched leaves at all. And what they found is that first Ivanovsky found that these material, when they crushed the leaves of them and passed them through a filter, the filtrate actually contained the infectious agent. And this was replicated in 1898 by the Bajering. We told this as a contagium vivum fluidum. This is a fluid which can actually cause the disease. The viruses were contained by the porcelain filter, but the filtrate contained the virus. And that was the first discovery of the virus. And now you can see that these are the cell culture material where you have a 0.2 micron filter, which can filter out the bacteria, but not the virus. Virus will pass through these filters as well. So this was the discovery. Then we will these are some of the very self-explanatory slides, and we will quickly go through these. So 1898 was the first animal virus that was discovered, and they had a key concept, not only small, but replicate only in the host and not the broth. And here you can see a timeline of the discovery. And again, you do not have to memorize those, and these viruses are not to scale, just to have an overview of that. First human viruses, when the Panama Canal was being made, a lot of people got infected. That was the discovery. Then this is the era of virology started more than a century ago. And the interesting part is that in the 1970s, the studies of the phages actually gave birth of molecular biology. So just to have an overview of an idea, no information. And you can see that Koch's introduction of efficient bacteriological method started the discovery of the bacteria, whereas the discovery of the electron microscope in the 1930s actually started the discovery of the viruses. And here you can see that some of these viruses, and we told you that the cartoon of this virus, bullet-shaped virus, this is a rabies virus that you can see, tobacco mosaic virus, bacteriophage. These are some of the pictures of the electron microscope with the negative stain that you have studied somewhere else as well. But this opened up the area of virology. And now you can see that the background is a cryo-electron micrograph, and then the modeled three-dimensional structure out of the cryo-electron micrograph. And you can just have a chemical formula of a virus. You know each and every protein, nucleic acid, or lipid present in a viral particle, and the numbers, you exactly know the numbers, so you can literally chemically write the formula of that as well. So this is 9:14. We will just stop right here, and we have just a few slides in here, so let's do this. So the virus classification is mainly on the nature sequence of the nucleic acid in the virion and the symmetry of the protein shell of the capsid, presence or absence of the lipid membrane or the envelope, dimension of the virion. These are the main components that we actually do discuss. So let's not rush. We just do have just 3 or 4 slides, but we will talk about this in the next class. Let's not rush. So we will stop here. So we will not still have our first quiz yet, because we have not finished that. But then we'll have our quiz. So I will just probably upload the quiz on Thursday to be finished on Sundays. So we will finish right there. And any questions so far? And we start with the virus classification that we left right here. And this is basically the virus classification that you already know. And that's the reason that I'm just not going into the detail of this one. Sorry about that. Detail of this one. The reason that this is, you all have taken 3050 microbiology, and there was a chapter there which was about the viruses. And in that, you have learned this. So you have learned the nature and sequences of nucleic acid, whether it's DNA or RNA. You have learned that the symmetry of the protein shell, the capsid. This is something we are going to talk in very detail, so we'll come to that. And also, there is another important thing, that there could be a lipid membrane outside the viral box, which is known as the capsid, and those are called the enveloped viruses. So just to recap the same thing, that if you just look at this virus right here, if you just follow my pointer, Is everybody with me? You guys okay with the PowerPoint slide?
Yes.
Okay. I'm just double checking that. Yeah, just put it in chat that if you have any difficulty on your side. So right here, if you look at this virus right here, this one as well, adenovirus right here, this small one, these are what we called as an icosahedral virus, and we'll come to that later. And if you just look at that, you have a solid protein shell in there. And these viruses are called the naked viruses, and they are not enveloped. But this one as well. Yeah, this looks like an envelope, but this is not. This is a naked virus as well. This is a Reoviridae family. But most of the other viruses that you're seeing right here, these one, you actually do have a membrane outside these protein coats. These protein coats in the naked viruses are also present right here. And outside that, you have a membrane and decorated with the same thing. And those are what we call as an enveloped virus. And based on those, we actually classified them. So what's the classification? You can just look at this ICTV online.org. This is actually the international organization which actually maintains the virus classification. Is extensive. You just do not have to get anything, but you can just see that how detailed it is. And you can just look at the coronavirus, like the coronavirus or the COVID-19 virus that caused by SARS-CoV-2. That's how it is named. And you will see that it is a beta coronavirus, and there are many others. There are alpha, beta, and gamma coronaviruses, and infects a whole lot of different animals, including the human beings. We actually do get other coronavirus infections, and that can give you just common cold-like symptom. This is very different, but this gives coronavirus infection is not new. This is extremely virulent in that sense. That is new, and it's under the beta coronavirus. You can just look at that how it is, just if you are interested. So the hierarchical system just looks like that how we actually do this for the organism. And again, this is where the controversy is that it is not an organism, and you really cannot classically do this, but there is some way we need to do this. So it is a kind of a pseudo-taxonomical way that we just use the organismal taxonomic properties to put it on the viruses. So what you're going to see that sometimes we use the order, and this will be, so suppose you have a name of the virus, and close to that, if it does have some property, you will have something like a viralis. Say, for an example, I'm just giving, we are talking about the Ebola virus. Now, Ebola virus do have an RNA genome. It's single-stranded RNA, and it is a negative sense. So you all know from your basic molecular biology that if I just take a DNA strand, it's 5' to 3'. The complementary is 3' to 5'. Whereas a negative strand RNA, instead of 5' to 3' will be 3' to 5'. And that's what an Ebola virus had. And that's why the order is known as, you do not have to memorize that, mononega viralis. Mono, single, nega, negative sense RNA, viralis is the order. And then the family is the Filoviridae. Filoviridae comes, this is a filamentous virus. You can see that it looks like a filamentous, and that's why it's called as a Filoviridae. Genus, Ebola virus, and this is the Zaire, where from it was isolated, and the species is Ebola virus. So when we just talk about the Ebola virus, we just talk about this Ebola virus, and there is really not a species that we can talk about the viruses. We just call its name itself, and then we talk about something else which is below the species level. What do we mean by the below the species level? Like, we can talk about an Ebola virus strains, either isolated from different parts of the world, which varies a little bit in nucleic acid sequence, or there are some different type of serological attributes that their proteins have been changed a little bit, which is also at the level of nucleic acid, but they due to that we can change those as such. Say, for an example, this is a very classic example. the influenza virus. So influenza virus, you already know very, very well. We talk about this H1N1, H5N1. There are many, many of these. There are two proteins, which is hemagglutinin and neuraminidase. And due to these combinations, we have many of these names. But these H1N1s, these are not species. These are just some of the proteins which are shuffled and reshuffled. And these classification they are all influenza virus. So we just stopped right here at the genus. We call that they're influenza virus. And then we actually call that is H1N1 or H5N1. And this is just due to their genetic makeup, which is below the species level. So this is an overview of that. That's about it. So now this is an interesting thing. And just for an information that once upon a time, the virus discovery was driven by the disease. Like say, for example, polio, smallpox, measles, mumps. These were all the debilitating disease. And all the scientists try to isolate the viruses to find what it is. That has completely been changed by this, what we call as a next generation sequencing, whereas you can just take any sample and what we call as a deep sequencing, you can find just a few, very few virus particles in your sample where you are sampling from, and you can just sequence their gene. And this is the way that is now the virus discoveries are being done. And there will be a saturation. At one point of time, you will see that probably everything that what we have in our environment, we have isolated that. So you can easily look at that. This is this is an analysis from 220 invertebrate species. So basically what they did, that they have taken this insect, crustacea, nematodes, mollusks, and these are all different Echinodermata, Tunicata. These are all different invertebrates. And from the samples that they have taken, either their body fluid or crushed them up, and then just took the whole extract of them, purified extract. And then from there, they have sequenced and found 1445 new viruses. Now, you can just go to your local, anywhere. You just go to a pond and just take a beaker full of water, and if you just send just a few milliliters of that water to a sequencing company, you may find that you have discovered a virus. And I'm just saying not all the time, but that is a possibility, and that's how the virus is now being discovered by this, what we call as a metagenomic analysis. And this is what is called as a deep sequencing. What it is, that these deep sequencing machines use a technique which we call as a next generation of DNA sequencing. And you can just look it out, next generation DNA sequencing. If you just wiki it out, you will be able to see that. And that the basic idea is that it can detect an extremely low amount of nucleic acid in the sample and almost sequence it, even if it is fragmented in different parts. It can those fragments can be aligned together to get the complete genome, and that's how the viruses are being now discovered. All right, so why do we care? And these are some of the things. The viruses outnumber the cellular life by at least 10:1. This is the greatest biodiversity on the earth, and the viruses drive the beneficial global biogeochemical cycles that we have understood. And these are the sources of new pathogen. Just think about that. Why do we care? We care because it just doesn't need any kind of explanation at this point of time that why our life has changed this year completely. And there were several outbreaks beforehand as well that you can easily remember. That 2013, 14, 15, there was an Ebola virus outbreak in Western Africa. Even there were many other than that. There is a Lassa fever, which is in the Nigeria area, Laos area in Africa, that's going on in there. But they are just small pockets. It doesn't become a pandemic. And that's why we need to be prepared for these kind of pandemics. And that's the reason that we just need to study this. And also another very important idea is there are many engineered viruses emerging as new nanomachines. And these are all used by mainly for targeting drugs in a cancer therapy. These are called oncolytic viruses. They either carry a drug to the tumor site, or they just by themselves go to the tumor and lyse them. And why it is so and how they can be targeted, you know that when you have a cancer where it is growing, they have extensive blood supply. So they have more vasculature compared to the other and faster cell growth. So preferentially, these viruses target those areas. The other cells may have, to some extent, some effect. This is the same thing that as a chemo. So the chemotherapy actually do cause some of the side toxicity. Engineered viruses which are being used also do have some, and that's the reason there is no viruses for even cancer, or the oncolytic viruses has been approved for usage yet. They are all at the research level at this point of time. Now, you may, of course, this is a huge debate about the whole thing, about the conspiracy theories about that the virus that has been made is made in the laboratory, engineered viruses, and it just came out. And at some point of time, once we go into the genetics of this, we will put that in the discussion about that. And just a preamble of that, the virus that is now the SARS-CoV-2, it is not an engineered virus. It did not come out of that. The thing is that why it is not, we will give you the idea of that it is just not possible. You can engineer that virus very easily, but how that combination of nucleotides can cause this extensive idea to manipulate 30,000 base pair by permutation and combinations, it is almost impossible to do this in the laboratory, because you cannot replicate these effects in a cell culture, whether it will work or not. You just cannot theoretically do that. And at that point, somebody is like, are you sure? It's probably done in the laboratory. But we will give you ample evidences that how the virologists think, and we will just argue on that. And you can argue on the other way as well. No, it is made in the laboratory. You can, of course, argue on that as well. And this will be a great discussion. And mainly this will be mandatory for the honors and the graduate students, but the whole class can take part in it. So I have also posted a discussion on this first part that will be mandatory for the honors and the graduate students. They had to post at least one post, but everybody can take part in it. The topic for that is that as we told, that the viruses are extremely small, whereas the mask that we are wearing, that can actually filter up. They are large enough to pass most of these virus particles, including the coronavirus particles. And still, why we think the mask is beneficial or how it does. So, again, you can look up the literature and post your positive thoughts of that. There is lots of scientific evidences. You can argue against as well. I mean, like if you see that, and that's the reason that everybody doesn't have to always agree to just one side. Then it's not a debate or a discussion. So just feel free to put your thoughts in there, and it will be interesting. Any questions for the first? Yeah, so we have the last slide. Sorry, I just always forget that we have the last slide, that there is an underlying simplicity and order to viruses because of the two simple facts. And this is what we are going to elaborate throughout this course, that all viral genomes are obligate molecular parasite. And you will see that I have used this term in your notes, and this is like a direct cut and paste from the textbook. And again, we can always argue that can only function after they replicate in a cell. So that's the important part. And all viruses must make the mRNA. And again, you know that making the RNA is the transcription mRNA. That can be translated by the host ribosomes. They're all parasites of the host protein synthesis machinery. So what happens that this is the most important thing. all viruses must make mRNA. That means that they need to transcribe their nucleic acid into the mRNA. The important concept that we will learn, DNA viruses make the mRNA, and that is transcription. Now, the RNA viruses will also make the mRNA, and this is RNA-dependent RNA transcription. So just not to confuse you that the transcription, what you have learned so far is from DNA to RNA. And here you will see that it will be from RNA to RNA as well for the RNA viruses. And we will call it transcription, but most of the time that will be making mRNA, you can argue, is a better term so that you don't confuse DNA to RNA transcription. Because the transcription from DNA to RNA not only make the messenger RNA, it also makes the ribosomal RNA by transcription. It also makes the tRNA or the transfer RNA by transcription. Some of the viruses make tRNAs or the transfer RNAs, but none of the viruses code in their RNA or DNA any ribosomal RNA. No ribosomal RNA can be made, so you cannot tell that it's a complete transcription. The ribosome is always stolen by the virus. And again, I'm using the anthropomorphic term, but it is stolen by the viral machinery, and they are the parasite of the host protein synthesis. So all the protein synthesized by the ribosome is actually it's used by the virus. Basically, it's exploiting, and that's a parasitic relationship. So it looks like a turkey, but it's not. This is a ribosome that is absolutely needed by the virus, and without an exception, has to be supplied by the host cell. So you have the large subunit and the small subunit. And again, remember that there are bacteriophages. They will utilize the bacterial ribosomes, which are different, 70S ribosomes with two different 30 and 50S subunit, whereas the eukaryotic ribosomes is also a little bit different. They're 80S with 40 and 60S subunits. And remember that the subunits are not additive.